Implementation of finite state machine comprising of DNA and DNA manipulating enzymes can be done as the following. The definition for this system, let's keep it like this. The symbols A, B and terminator T are implemented as a sequence of 6 specified nucleotides. The state S1 or S0 of the machine is defined by a 5 overhang consisting of 4 specific nucleotides. The terminator T defines the final symbol read. The machine consists of an input molecule, an output detector, and 2 enzymes, FOKL and ligase. The input molecule consists of a FOKL recognition site F and a spacer X, where the number of nucleotides are defined. A nucleotide sequence defining A and B, and a sequence with the remaining symbols, and the terminator sequence. FOKL is a restriction endonuclease which can bind to F. It cleaves the DNA on the sense strand 9 nucleotides downstream and the antisense strand 13 nucleotides upstream of the nearest nucleotide of the recognition site. Thus, the space X defines where the FOKL is cutting, and the cleavage in the input molecules result in the first intermediate state SO, 5 overhang. Reading A. Ligase litigate this product with the transition molecule. This transition molecule determines the transition between the states in this example. If A is read move from SO to SO, the other transition molecules can be generated defining all other possible transition rules. These molecules are designed such that the 4 bases along 3-inch overhang read the symbol. The spacer defines the cutting point of FOKL1 and the state the machine will transit to. The input molecule and the transition molecules get litigated. A new digestion with the FOKL leaves a 5-inch overhang representing SO and reading A and B. This cycle continues until all the remaining symbols are read and state transitions are executed. The last digestion leaves a 5-inch overhang with the terminator T, sequence defining thus final state, in general either S0 or S1. In this example, we have S0. The molecule in its final state gets litigated to an output detector engineered to recognize either states S0 or S1. This forms an output reporting molecule which can be generated by gel electrophoresis. And let's now know about memory unit. A computer stores its information in the memory unit, and all the transaction related to the transfer of information or instructions is done there. As a human being, we remember things that are going around us in the brain. A recent study has suggested that there is an incredible characteristic of DNA is that it can store huge amounts of data, theoretically up to 2 bytes per nucleotide or 455 exabytes per gram of DNA. An important feature of DNA is the relatively permanence of storage. Even after the cells die, one might be able to recover the information from DNA. Another study engineered an integrated logic and memory device where the memory is raised from the ability of recombinases to write information in DNA. A simplified diagram of the modular memory device, which is a transcriptionally controlled network composed of 2 transcription factor encoding genes, a sensor gene and a positive auto feedback gene. This pGAL molecule is having GAL 1 by 10 promoter, and this CYC has a CYC1 promoter. DNA-BD is the sequence encoding a DNA binding domain of respective transcription factor. The network can be in 3 states: off state, on state, and in the memory states. If the system is in off state, it has never been exposed to a signal. Here we have a galactose signal for testing. It is on if the galactose is present. In this case, the signal induces the synthesis of transcription factor, the sensor. This triggers the expression of another transcription factor able to bind to its own promoter. The system is in memory state. If the signal is removed, the autofeedback activator is able to initiate its own expression even if the inducing signal is lacking, which means that the system has stored in which means a rewritable recombinase addressable data module is able to store data with the DNA sequence. Serine integrase and excisionase are used to invert and restore specific DNA sequences. The system has two potential inputs, set and reset transcription signal. The set signal drives expression of integrase, which inverts the DNA element functioning as a genetic data register. Flipping the register converts the flanking sites, here which are shown in the triangular shape. The system is now in state 1 or S1. Alternatively, a reset signal drives integrase and excisionase expression and restores register orientation and the flanking sites. The system is in its other state S0. The register comprises a promoter, which is the driving state dependent and strand specific transcription of either red or green fluorescent protein, which are the two possible outputs of the system. In this way, these are set. When it is in the case of buses, a normal silicon processor consists of wires, as we know before. But when it comes to biological microprocessors, wires from silicon processors are made from solid and state metals, whereas in the biocomputers, systems engineered so fast consisting of signaling molecules such as regulatory proteins. For example, in the construction of a XOR gate, as we seen before, in biological ALU, in this case, chemical signal molecules used as wires between different logic gates. Here we can see the regulatory proteins of cell type A, which is helping them in converting to cell type B. Coming to the applications, we know that biological microprocessor exist, but we didn't see anywhere which in the real world. So, but let us know where they are used at least. They are used in disease diagnostics. It is used in detecting multiple disease indicators such as mRNA of disease-related genes associated with small cell lung cancer and prostate cancer. It is also used in environmental monitoring, which is another important application. A cell-based biosensor using logic gates has been used to detect arsenic, mercury, and copper ion levels in the atmosphere. Tissue engineering and tissue regeneration is also an application of this. Furthermore, biocomputers can be used to engineer context-dependent programmable drugs. A biocomputer with a context sensing mechanism which can simultaneously sense different types of molecules has been engineered. Biological microprocessors can potentially be used to control development and cell differentiation and reprogramming. All these processes depend upon gene regulatory networks. There are some other applications also which include drug screening. They can be used for control of biological systems, metabolic engineering. In future, it might be used to detect a broad range of molecular disease symptoms and react with the release of drug molecules suitable for the treatment of specific condition. Another important application is in the area of control of malign growth. Some interesting experiments with logic-based biological devices have been executed to detect cancer cells and used to induce selective apoptosis of these cells. Here, cancer cells may include small cell lungs, prostate, or HeLa. So, this area of controlling the malign growth is really important for the cancer patients. So, hope the experiments go in a right way. And the future scope, if we discuss about future scope, we can see so much of importance in the present experiments that are going on. It includes there can be devices which can include sensors that can swim inside the arteries, detecting the buildup of formal plaque and rapidly delivering medications to the affected area. And some other experiment, like if there is a sensors that can detect and destroy the cancer cells inside the body, and pollution monitors that can be deployed in the environment, detecting and utilizing dangerous toxins such as arsenic can also be developed. Previous research only proved biological gates could be made, but their advantage of biological gates over previous attempt is that they behave more like their electronic counterparts. So this can be seen as a replacement of the electronic logic gates that are present now. New biological gates are also modular, if, which means that they can be fitted together to make different types of logic gates, paving the way for more complex biological microprocessors to be built in the future. Um, there are certain challenges, or let's look about them. Challenges faced are finding a way to link multiple biological gates, similar to the way in which electronic logic gates are linked together for enabling complex processing to be carried out. Biochemical reactions have by nature often long reaction times. The input and output signals are of analog and not digital by nature. Biochemical reactions are often in solution and not in all classes compartmentalized, which results in the lack of signal separation. And there are also like techniques that reduce error rates have to be developed. Handling fragile DNA molecules during manufacture of biochips is difficult. If DNA computers are forced to operate serially, their performance becomes really poor. This is because when too much of data is fed into the DNA, then it might affect the performance. So this is about microprocessors. Thank you.